Winery recently bought a state-of-the-art machine to monitor and control fermentation by measuring redox potential. The winery decided to monitor redox potential at three different stages: alcoholic fermentation, bottling phase, and wine storage phase to ensure production of high-quality wines without faults. After 4 months of the purchase, the team decided to carry out a sensory tasting session to check the quality of the wine and the efficiency of the new machine. During the session, the wine was found to taste pickly and bitter. So the fermentation expert was called upon to check if defect had anything to do with the new machine, and if not, to figure out what could have caused this. So the problem setting here is a winery bought a new machine to monitor and control the fermentation process. When a tasting session was carried out after 4 months of purchase, the wine was found to taste pickly and bitter. The fermentation expert knew that it could be potentially lactic acid bacterial contamination in wine that could have led to wine becoming pickly and bitter. So when we talk about wine, high quality wines essentially have protection against oxidation and have a reducing character. Therefore, it's essential to measure redox potential in wine as it helps us understand the oxidation and reduction state of wine. It also helps control and monitor the fermentation process depending on what is the oxidation reduction state of wine, and ultimately contributes to quality, taste, and aroma of the wine. During wine production, redox potential is continuously monitored to know when ethanol production begins. And why is this essential to know? Because you get to know once ethanol is produced, you get to know that your microbe, which is yeast Saccharomyces cerevisiae, is active. Grape juice has a redox potential of around plus 300 to plus 350 millivolt. When the redox potential changes to minus 250 millivolt, then it is indicative of start of ethanol production. And when the redox potential is at a minimum, you have maximum biomass concentration. And further reduction in redox potential, it blocks yeast's metabolism and inhibits growth of any other microorganisms. Yeast typically act when the redox potential is in between plus 350 millivolt to minus 200 millivolt. Above 350 millivolt, oxygen becomes toxic and inhibitory, and below minus 240 millivolt, concentration of dissolved oxygen is so low to support any sort of microbial growth. So you would have gotten an idea why is it important to monitor redox potential. So what is this redox process or oxidation reduction process? Redox indicates oxidation and reduction reactions. The growth of any microbe in food depends on several factors, okay, like pH, temperature, redox potential. As chemicals present in food can act as both electron donors and electron acceptors. So oxidation means a tendency to lose electron, and reduction means a tendency to gain electron. Therefore, if food has more oxidizing compounds, then the redox potential will be higher. And when there are more reducing compounds, the redox potential will be lower. Different microbes require different levels of oxygen for their growth. And the level of oxygen for growth is determined by redox potential. Based on redox potential, microbes can be classified as following. If you have high redox potential, it means that your medium has oxygen. It supports growth of aerobic bacteria or aerobic organisms. If the redox potential is low, it means your medium does not have oxygen. It is deficient in oxygen, meaning support of anaerobic microbe is favored. Then you have facultative anaerobes, which can grow in presence or absence of oxygen. So really, it can grow when the redox potential is high or low. Finally, you have something called as microaerophilic, which grows in very limited amount of oxygen. So it can grow in medium containing very low redox potential. Now, if you understand this, you would know that One of the ways to preserve food is to play with this redox potential, be it wine or any other type of food. And lactic acid bacteria spoilage in wine typically occurs when the redox potential value is below minus 200 millivolt. The redox potential changes from minus 200 millivolt to minus 500 millivolt when there is excess lactic acid bacteria in your wine. So how can we modify the redox potential in order to have control on fermentation process? Lactic acid bacteria is known to cause something called as malolactic fermentation, which is occurring after ethanol production. So this occurs typically in your red wine. In order to enhance the flavor of red wine, lactic acid bacteria converts malic acid to lactic acid. And how can this be controlled? Essentially, we have additives like nitrite and you have vitamin C. If oxygen is replaced with nitrite or vitamin C, they compete with oxygen for electron. Nitrite can accept electrons more readily than oxygen and thereby help in shelf life extension. Second is packaging. Packaging is often function of oxygen permeability. So what kind of package we choose is very important. If the package facilitates oxygen to enter, then the redox potential is going to be higher. And if it is higher, it will facilitate growth of aerob. Lactic acid bacteria as such is microaerophilic. So even in presence of minute quantities of oxygen, it can grow. Finally, it depends on the structure of food. What I mean by structure of food is discrepancy in the reception of air. Suppose we have fresh fruits, vegetables and meat. Okay, on the inner surface, the redox potential is generally lower because there is no oxygen received on the inner surface. So it will facilitate growth of anaerobe. On the surface where there is enough oxygen being received, it will support growth of aerobic organism. So from this conversation, you know that redox potential is an important parameter that can be used to control microbial growth in several food products, right from fresh vegetables to fresh fruits, meat, and your wine.